For our next presenter, we have Josh Levy, who is a project scientist at Scripps Research.
Yes. Which microphone? Clicker. Yes. Okay. Right. I'll turn it off. Just. Yes. All right. Hopefully you can all hear me. So today I'll talk a little bit about some of the wastewater surveillance work that we've been doing, starting from sort of basic technology development all the way up into sort of deployment in a global sort of sentinel system for wastewater clinical integrated surveillance. So first I'll talk a little bit about sort of the basics of how we go about looking at an emerging outbreak or really any disease spread and trying to understand what we're seeing, what we're not. And usually the basic tool we use there is clinical surveillance. We have some index case or sampling individual cases from in that outbreak. And a lot of those go missing. You know, we can't detect everything usually. And we can infer individual cases that might have happened in between things that we do detect. But there's a lot that sort of goes missing. And we get this sort of initial sort of epi curve of initial cases, but we don't always know how much isn't there. We might be able to rely on something like sequencing to infer some of these individual nodes, but there's a lot that's sort of in that mix that we kind of miss. And there's been work that's been done trying to understand if there is this sort of delay, this limited sequencing of what we're actually looking at, how does that impact our ability to identify novel lineages, things that might be expanding and have significant public health impacts? So this plot, suffice to say, basically shows that as you decrease the rate of sequencing among cases that are being sampled, you lead to an increase in the amount of time to actually detecting a novel lineage. So this can have significant public health impacts. We'll talk about specific cases about that in a bit. One thing that's very important to note is that SARS-2 is sort of an isolated example where we were really sequencing a lot of cases in a lot of the world. And of course, there is a lot of the world that very little actually does get sequenced. And this leads to our ability to, our lack of ability to be able to identify things that may actually lead to large outbreaks, potential impacts to public health. You can see this is from here in Washington State. It's a little bit out of date now, but this is the last time that they were putting numbers up as to how often they were sequencing. You can see that during the main parts of the pandemic, there were very high sequencing rates, lots of sequences being generated, and by the end of 2023, that sort of peters off, and there's very little being sequenced. But as an alternative or as a supplement to this, we also have wastewater, and wastewater is a pretty powerful tool for public health. It really closely provides another analog for case or burden in a particular area. So you can see this is from San Diego, where I work, and you can see that the case and the wastewater viral load trajectories really kind of are highly correlated, tending to see a little bit of a leading signal in wastewater. And this is what it looks like when you're just measuring the concentration of virus in the wastewater. But what we wanted to do, and where things sort of started for us, was to get into how do we actually meaningfully interpret sequencing of wastewater. So in order to do that, we first had to justify it. And we noticed that wastewater as a surveillance modality was incredibly cost efficient and incredibly capable of surveilling a large number of people, sort of without any particular sampling bias. You didn't have to, swab individual people's noses, and as a result, you're able to basically passively surveil an entire community all in one go. And this sort of comprehensive, cost-efficient nature means that it can be done sort of, at scale with limited financial implications. And this last bit here, this ability to potentially sequence this is what really got us excited, because we wanted to be able to understand what was circulating, what lineages of the virus were circulating here, speaking about SARS-CoV-2. The thing is if we want to use our traditional tools for SARS-CoV-2, they don't really work very well for being able to extract, consensus genomes out of a wastewater sample. They're highly mixed, and it's sort of an ill-posed problem to be able to do this. So you could have one potential haplotype that you could be inferring, but also those same reads could potentially, you can't really see because my text is covering it, but multiple different haplotypes can be basically all encased within a single sample, and it's impossible to identify which mutations are associated with which particular virus genome. So what we did is we put together a method. I'll give a brief little summary of it because we're talking more about the applications of it today. But basically, we encode individual virus lineages as rows within a large matrix based on the mutations that they contain. So each lineage is encoded by a unique mutational barcode, and each individual sample on the top right is encoded in terms of the individual mutations that are in that sample and the frequency with which we find them in our sequencing data. And we can put this all into a mathematical framework, which we now call Freya, that allows you to infer the frequency of the individual lineages that are circulating in the community. And just for a sense as to what this looked like when we were sort of tracking the Omicron outbreak in San Diego, we're able to pull in individual lineage frequencies and directly incorporate things like the viral loads to get a sense as the actual contributions of these individual lineages in time. Here we see BA1 and then BA1.1 sort of take off, and really delta associated diversity remains, but just remains sort of at low levels. So I want to talk a little bit about how we bring all of this together with clinical data, with epidemiological data, into sort of an integrated picture of how we do public health surveillance. So here I'm focused on an example town with 4 wastewater treatment plants. And from a standard sort of epidemiological clinical perspective, we might be picking up some cases in different parts of the cities. And we could also have a wastewater signal. We might have individual regions where we're starting to see higher concentrations of the pathogen, and others where we don't necessarily see it. And we can sort of like bring these together to have sort of a unified picture of what matching clinical and wastewater surveillance should look like. But then we want to bring in this genomics question. Of course, we have sequencing of individual cases, each associated with individual lineages. We can also then use a method like Freya, which again, you can't see too well because of the text, but to be able to pull the frequencies of the individual lineages within each of these different wastewater catchment regions. And then lastly, you can sort of bring all of that together to get this sort of combined epidemiological genomic picture through both clinical and wastewater. And that allows you to have this sort of redundancy that's actually really important, because you can see that there's things that don't quite match up. You'll see that, yeah, maybe you see clinical cases of, the yellow variant in one side, but you don't actually see it in the wastewater, or vice versa. You see in clinical, you see no blue variant, but you see lots of it in the wastewater. And this can highlight different regions that are not being very well serviced or surveilled by a clinical surveillance, for example. So we want to focus in on this highly integrated picture. And we also have developed another sort of platform called outbreak.info, where we're taking this sort of information. And this sort of was our next step of sort of bringing in broad clinical information, epidemiological information, and wastewater, both viral load and genomic sequencing information, in order to sort of bring together this sort of unified hub for outbreak intelligence. And of course, after that, we wanted to be able to make this accessible to users. So we also have built and assembled some APIs, web clients, things like that for people to be able to access this. But what can you actually do with this sort of stuff once you've taken lots and lots of data from public repositories, working with collaborators, in order to sort of centralize and sort of derive insights from this data? So as an example of what this looks like, I'll show for California, where our research institute is based, using both data that our lab has generated as well as from lots of other data generators across California, many of which are associated with the CDC's National Wastewater Surveillance System. And we are able to pick up both, these sort of viral loads from wastewater and clinical pictures, where you can basically see, these two are very highly correlated again. again, you can't totally see, but the clinical curve basically is nicely correlated here with the wastewater curve. And we can aggregate across the state to pull in all of these different wastewater plants and all of the clinical data across the state. But we can also pull in sequencing information from all of these. We can see as different mutations rise in frequency and drop in frequency over time. And that foreshadows changes in the lineage landscape in the community or across the state. So here you can see sort of highly analogous wastewater and clinical curves. And then we can start looking using our Freya approach to pull out the individual lineages that may be responsible for this. So you can see on the bottom right, there's sort of this rise of JN.1. This is from this past fall, basically as that was sort of ticking up across the US. And we can sort of look at these, one next to the other, and we can see very similar pictures in many regards. And as I mentioned, they're highly correlated sorts of information modalities. But what we do see is that wastewater tends to be a little bit earlier on average, although this is not always the case. So to further deploy this in sort of a larger global setting, we worked with a lot of collaborators, many of which are using Freya, and we've been in discussion with for the last few years, basically trying to test out this sort of approach across different research institutes across the world to see how well this plays. And the text is sort of covering it at the moment, but each of these groups is using kind of widely different sorts of approaches for doing the surveillance. Some groups are using spike-focused sequencing, sub-spike only sequencing, nanopore sequencing, Illumina, all sorts of different platforms. And they're able to do this sort of surveillance and get similar sorts of results regardless of the platform and sort of the nature of the surveillance system. So throwing up a few sort of examples here from this program, and there are little gaps associated with some of these programs having lapses in basically government funding switching over, different programs basically having switches between locations, things like that. What we can see is that we're able to track these sorts of lineage dynamics over time, and that they're not all happening at the same time. So you can see certain lineages might be popping up earlier in the US versus South Africa, in Spain versus Denmark, etc. And we're able to pull all this information and do some sort of more systematic characterization of what might be going on here. In general, we see that these signals are highly correlated. On the left-hand side, we see really high correlation coefficients associated with the actual variant breakdown across these different surveillance programs with respect to clinical data. And also we see slight biasing, about 10 to 15 days or so, generally in wastewater ahead of clinical surveillance. So you can see these are detection dates on the right-hand side. And in general, not everything is always detected at first in wastewater, but in general, things are right-shifted, suggesting that wastewater is generally finding things ahead of clinical surveillance. And so what does this look like in an outbreak survey or in a regional spread scenario? Here, I can't see too much because of the text, but basically on the bottom here, we're looking at an outbreak scenario in sort of Southern Europe, Northern Africa region. And this is a BQ.1, which didn't originate in this area, but was spreading throughout the region. And we see it first pop up in Algeria, and you then expect, okay, this is a highly fit variant. It may start propagating throughout the region. Where is it going to be expected next? And you can see sort of on the bottom here that the wastewater picture, and this is looking through our Spanish Catalonian collaborator, picks up data in Spain about 10, 15 days ahead of clinical surveillance anywhere in Catalonia or in Spain. In fact, much earlier than a lot of other places in the region that are much more connected to Algeria. So if you actually look in mobility data, you can see that actually Algeria and Spain are actually not very connected. Despite their very close proximity, there's not many flights that will take you from Algeria to Spain. Whereas there are a lot of flights that take you from Algeria to France, and France and Spain are actually quite connected. So you're able to start thinking a little bit about how we expect this outbreak to play out and start responding in kind to be able to make that happen, or to circumvent undue spread. Another very useful case is not just in spread of ongoing lineages or of already circulating lineages, but of lineage emergence. So through our collaboration at NICD in South Africa, we actually are able to detect, or were able to detect, the first known detections of BA286 in advance of clinical data. Of course, this was not available until after, so these are date matched. The actual initial detections of BA286 were actually out of Denmark instead. But the actual dated earliest detections are in wastewater and then in clinical data in South Africa. And we can see that wastewater provides this early kind of leading curve ahead of clinical surveillance for the BA286 outbreak in South Africa. But mobility data tells us a different story here. Here, we're not able to really get much information as to where BA286 originated from. We know that in Southern Africa, as Bethany was talking about earlier, there's clustering of mobility and travel across the region, but it's without any more access to see what's happening in those public health or in those communities. We have no ability to figure out where actually that might be able to originate, but we can better date exactly when those outbreaks are happening via wastewater. And wastewater may be an easier way to start bringing up public health capacity in many of the communities where we currently have somewhat of a blind spot for public health surveillance. And focusing in back a little bit on sort of international, zooming in into what we can look at, we can pick out sort of these detailed pictures of what's happening over time as a lineage emerges. Here's looking at BA5 across the US. You see basically similar but not exactly matching pictures of what is happening in wastewater versus clinical spread. And I think the most important sort of thing to get out of this is that it's actually much more important to sort of see these as sort of two parts of a larger whole. That some of these sites where you see sort of the hashing you're seeing lineages detected at significant prevalence earlier in wastewater, but other sites you see it happening earlier in clinical. And bringing all that information together allows us to have a lot more information about what's going on. And when we deal with other pathogens or later periods of this ongoing pandemic where we have much less sequencing data available, things tend to shift to larger gaps in between wastewater, earlier detections, and clinical. And this is a huge point. On the right-hand side, we see sequencing of SARS-CoV-2. In comparison, flu, one of the most highly sequenced viruses out there, is very rarely sequenced in comparison. And this problem only gets, more significant as you consider the level of sequencing that happens in lower and middle income countries. So I think wastewater provides a great sort of platform for enriching and expanding our surveillance capabilities. And we have Freya functionality to be able to do this for a variety of pathogens now. And with that, I will take any questions you might have. This is an effort across many labs across many countries. I can't name everyone here, but I've tried to highlight some of the most excellent collaborators that I've worked with. Excellent. Thank you.
Thanks so much, Josh. I will pass the mic around for questions.
Thank you very much. A wonderful presentation, and you present wastewater sampling as an important tool. But my question is, so among, I was a little confused because you present data to show that wastewater is faster than clinical sampling, but actually I think that you provided only one example where wastewater found the lineage first. So I'm interested to know what your definition is in terms of your definitions are there. But then related to that, given that strong correlation between wastewater sampling and clinical sampling, can you tell us what the sensitivity is given a prevalence in the population? So what prevalence do you need in a population to detect it?
Yeah. So I mean, the only real case study that I showed you was the BA286, where we had this early detection. I should say in San Diego, where we've probably done our most complete vetting of this, we almost always see it earlier in wastewater, usually 10 to 14 days. And we saw that in the paper that I sort of mentioned there, which I didn't get into too much detail because I wanted to look more into sort of the larger scale application. We see regularly for Delta, Alpha, Omicron, now for BA286 as well in San Diego. We saw earlier detections, again, this sort of 10 to 15 day window regularly. This further expands when you look into places with less clinical surveillance. So places like Bangalore, for example, we had some cases where we're looking at 100 days or more of earlier detection in wastewater ahead of clinical. In terms of the sensitivity that you asked about, though, wastewater can be incredibly sensitive. Talked a little bit at the end about Mpox. During the clade 2 outbreak that happened across, well, globally, but in our own backyard, we were able to detect Mpox, and quantify the levels of it when there were only, a few cases per day in the region. So suggests that even a very small number of people infected can lead to a detectable signal. One thing that we really need to work on, though, as a community, is the tools to actually enrich for viruses and pathogens of interest that make these sorts of methods more sensitive, and the actual sampling methods that we go about that actually allow us to have a more wide net. So, whether we're doing a grab sample versus a composite sample versus a passive sample, that has different implications for the sensitivity of your measurement.
Thanks so much.
Thank you for that great presentation. I have a question on how, if your group has talked at all about like disentangling human contributions, because like in North Carolina, we had a signal for H5N1 and it turned out that they were dumping raw milk and then that was contributing to wastewater. So just how your group has kind of been thinking about that.
Yeah, so it's a great point and very important for things like H5N1, which we've done a bunch of milk analyses about or of using Freya. But in terms of understanding the contributions, I think this is still kind of an open question. There were some very famous examples during or in New York City, looking at sort of cryptic variation, and they were measuring like rodent DNA also in the wastewater. And there was suggestion that actually these were being, given off by rodents. I think that that's a sort of a misleading interpretation. I think for H5N1 in particular, you need to be able to sort of evaluate the information on the ground. This is, it's not a virus that we're going to see large amounts of asymptomatic transmission going on for. And odds are that you're probably going to be seeing more milk associated detections via wastewater than not. But that has to be, that has to dictate how we choose to use that information. I think everything needs to have an appropriate, prior or asterisk associated with it to be able to, push stakeholders, politicians, et cetera, in the right directions and give them the information they need.
Yeah, great talk, Josh. Let's say we had a variant that we really wanted to stop the spread of. Do you think that, given what we know of connectivity data, that we can actually inform the turnaround time that we need in labs to actually detect something from wastewater before it starts to affect, I guess, public health?
So I think it's not an easy in practice. Actually, stopping the spread is wrought with all sorts of socio-political implications. But in principle, if you start detecting something in wastewater, and pretty regularly in wastewater from across the US, for example, we're picking up all sorts of weird cryptic variant signatures. It's hard to know how to place, is this the next most fit variant that's going to storm the world? Or is it just sort of somebody who's harboring a chronic infection? But I do think that the key thing here that wastewater offers is that it's sufficiently low cost and passive that you can, at a relatively low cost for a city anywhere in the world, have regular surveillance that doesn't need to be spun up or spun down. You can have many pathogens being tracked at once and then respond in kind. You don't necessarily need to bring down the turnaround times of this or that. Whereas for clinical, you really may need to scale up to huge numbers of cases that you're being able to, or that you're detecting in order to identify that one case associated with that one variant. So I think that's part of the power of wastewater, is that it's passive but still very actionable, and still very timely, because you can do it that way.
Last question.
Thank you for the great presentation. I had a question. I'm going to take us back to the leading indicator, wastewater surveillance being leading indicator. I thought it was really interesting because by the time patients shed and sample is collected and then you sequence, that gives you a signal quicker than case surveillance data. That would make sense in places with health infrastructure with not so much surveillance reporting capacity. But in a place like San Diego, all of that took sooner for decision makers to see than aggregated surveillance data. So the lag is probably driven by, is it the reporting or aggregation of data? Just wanted to see if you have additional comments.
Yeah, so that's something that comes from a few different factors. Some of it's just asymptomatic transmission, right? People will still shed if they're asymptomatic. And then some of it is just how long people go until they test, report, etc. So those sorts of gaps are what sort of dictate that in San Diego. But that's not to say that this doesn't work in places where you don't have the same level of sewage infrastructure. We've done this in numerous different cases, leveraging informal sort of sewage disposal and transport, or just existing canals, waterways that have waste that pool into them. And if you're, strategic about how you go about it, and we are fortunate to work with some great geospatial modelers that allow us to sort of get a sense as to where waste may be draining in a community. We can find specific sites, say in a canal or in a pond where something's draining into, that are very useful for wastewater surveillance. So those are places, again, where you might have much less reporting, and that leads to even larger gaps in between wastewater and clinical detections.
Awesome. Let's all thank Josh one more time. Thanks so much for the presentation.